Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
Good evening. I'm Yvonne Stapp, and I welcome you to Contemporary Science Issues and Innovations. Tonight, we welcome one of the great scientists of our time, Dr. David Page, Director of the Whitehead Institute for Biomedical Research at MIT, Professor of Biology at MIT, and investigator at the Howard Hughes Medical Institute. Dr. Page became a science star very early in his career. He received his MD from Harvard in 1984 while working in the MIT lab of a leader in the Human Genome Project. He subsequently joined the Whitehead Institute and MIT faculty in 1988. Dr. Page's lab achieved early distinction by mapping the Y chromosome in 1992 and then publishing the complete sequence of the Y chromosome in 2003 with the collaboration of Washington University Genome Sequencing Center. In 2005, he was appointed director of the Whitehead Institute. Dr. Page founded the Whitehead Task Force on Genetics and Public Policy, and he serves as co-editor and associate editor, respectively, of the two major journals in the field of genetics. He's a member of the National Academy of Sciences and also the Institute of Medicine. Dr. Page has received a long list of distinguished awards, several of which we've listed on the Science for the Public website for this event. But we recommend also that you visit the Whitehead website to see more details of Dr. Page's outstanding contributions to genetic science. Tonight, Dr. Page will focus on several points about the Y chromosome. First, its evolution. And second, why its diminutive size and the concern among experts for a long time that maleness would disappear. And third, the relationship between the sex chromosomes and some diseases and conditions that disproportionately affect either males or females. It is a very great honor and pleasure to welcome Dr. David Page. Welcome, sir.
Thank you very much.
And I'd like to start with the evolution of the Y chromosome. Where did it come from?
The X and the Y chromosomes are usually thought of as completely separate, but it turns out that 300 million years ago, when we were reptiles,
believe it or not.
Yes. Our X and Y chromosomes had their beginnings as a perfectly identical pair. And so, in fact, what live on today in us as the X and the Y chromosomes were once upon a time exactly the same of a member. They were members of an identical pair of chromosomes.
Okay, all right. Now, so why did they?
What was the
how
path?
Did the male? Right. Yes, exactly. What was the payoff?
Well, so it turns out that back when we had no X and Y chromosomes, 300 million years ago, our ancestors I'm referring to, our sex was probably determined by the temperature at which we as an egg incubated. This is true today. Turtles, crocodiles,
exactly.
Alligators today.
Yeah, okay.
So, but what happened then? So at that time, whether we developed as a male or a female was determined by an environmental cue. Almost, you could say, in software. But what we then did was to move sex determination into hardware, and a mutation arose on what had been a perfectly ordinary chromosome to begin to turn it into a Y chromosome.
Okay.
And so this chromosome now took on a sex-determining role, a male-determining role, that it hadn't had before. That was the beginning of what has been a 200 or 300 million year saga, a journey of this ordinary, unsuspecting pair of chromosomes to become today's X and Y. Along the way, to sort of cut to the chase, it looks like the X chromosome retained most of the gene content It retained most of the innards of that ancestral chromosome that gave rise to the X and Y, whereas the Y chromosome lost almost all of the genes that it once shared with the X chromosome.
Okay. Was there anything peculiar in that split once you had sexual reproduction, this new sort of thing? So you lost some genes in there. Was there anything else in there we should know about? Goodbye, male chromosome.
Well, so first of all, I want to make clear that before we had X and Y chromosomes, we still existed as males before that time. Our ancestors existed as males and females. The males made sperm, the females made eggs. We were reproducing sexually. And so it turns out, and I think people often get this point very confused, that it turns out the sex chromosomes were a late addition in evolutionary terms to a perfectly functioning system of sex determination and sexual reproduction. It is never taught this way today in the textbooks. The textbook always begins with the X and Y chromosomes. But it's important to understand that, in fact, the X and Y chromosomes were a late addition to an already nicely tuned functioning system.
So we had sexual reproduction, but it was a little different from what it is. But can you just step back for 1 second there? In terms of sexual reproduction, was there an advantage or disadvantage? Why would nature go in this direction? Why not just clone ourselves endlessly? It worked for bacteria.
Right. So there are a whole bunch of questions in there. Let's just tease a couple of them apart. So one is, why reproduce sexually when we could just reproduce clonally? Or another way of framing that question is, why bother to have males? Because, because basically, if you reproduce clonally, then everybody can have offspring. If you have males and females, well, it's only the females that actually give birth. The males are just coming along as a source of a little bit of genetic variation.
Oh, a little.
And maybe some trouble along the way. It makes life much more complicated. But no, seriously, people think that it is that what males or sexual reproduction adds to the picture is it creates an opportunity to sort of mix and match genes over the generations. And we often think of this as being a source of evolutionary progress, but it also turns out it allows organisms to not fall behind. So So it turns out that during the course of, as genes are passed down through the generations, they often sustain little damaging mutations.
Yes.
Okay? And with sexual reproduction, it is possible to swap out or purge a damaged copy of a gene and essentially replace it with a good copy. And in fact, it is this problem of the need to purge genes of mildly deleterious bits of damage to them. It was the inability of the Y chromosome, as it began to part ways with the X chromosome, that led to the demise of so many of its genes. So in fact, the Y chromosome, despite its being called a sex chromosome, is actually transmitted in an asexual or clonal fashion. So my Y chromosome is from my father, from his father, his father, his father, his father. Unlike any of our other chromosomes, which are sort of a mishmashed mix of 4 grandparents, 8 great-grandparents, and so on.
The whole tribe.
The whole tribe, all the way back there.
Yeah, right.
And so it was actually the Y chromosomes going it alone that led to the demise of so many of the genes that it carried.
I see. Okay, now we understand that the Y became very little. But one of the big issues was, why did that happen, and where did those genes go? And you were a major source of what happened to that. Okay.
So we've had a lot of fun reconstructing how did these 200, 300 million years play out with the X and the Y chromosomes. So first of all, part of the reason that those genes could disappear from the Y is because they were retained by the X. So I don't want to anthropomorphize too much, but the X chromosome seems to have had this better,
efficient.
Well, it was more efficient at retaining. It maybe was a better nurturer of these genes once shared with the Y chromosome. Yeah. So in some sense, and since everybody has an X chromosome, males have an X, females have two X's. Since everybody has an X chromosome, it meant that everyone had at least one good copy of these genes.
Okay.
So it sort of took the pressure off the Y to provide those functions. And so along the way, the Y chromosome lost a lot of genes. They were retained by the X. And then the Y started developing some other specialties. It actually not only lost genes, but especially during the last 30 or 40 years of primate evolution, the Y chromosome has actually gotten in the business of importing new genes to its shores. And these have actually helped drive the Y chromosome in our species and in other primates towards a very strongly specialized role in sperm production. So that male fertility today depends to a very significant degree on genes on the Y chromosome, many of which have been acquired by the Y chromosome during the last 30, 40 million years of evolution.
That's rather rapid, isn't it? You know, so
and this actually flew in the face. There had been a lot of ideas saying, "Oh my gosh, if the Y chromosome has gone its own way, this helps us not only understand that the Y chromosome is going to lose genes. It helps us understand that loss of genes, but it came as a complete surprise to us and the scientific community that." Despite this going it alone path, that the Y chromosome was in occasional communication with other chromosomes and occasionally actually acquiring a bit.
Right, imperialism, right?
Well, perhaps a little bit of importing, right.
But I think in the scientific community, you know, well-respected scientists were concerned about this diminished thing, which they assume was happening like every other week or something, or rather, it appeared, but you said, no, it happened much earlier.
Well,
how did that come about?
So there's been, for decades now, there's been a debate about the past of the Y chromosome and also predictions of its future.
Right.
And so I've often said I've spent the better part of my career defending the honor of the Y chromosome in the face of many insults to its character, but especially to its future prospects.
Yeah.
And so the question was, yes, the Y chromosome had lost so many genes that it once shared with the X chromosome, but could we actually reconstruct the trajectory of that loss? And so about 10 years ago or 12 years ago, it was actually editorialized in the pages of a scientific journal of some repute called Nature. That it was editorialized that the Y chromosome would, at its current rate of gene loss, disappear in 10 million years. And, you know, given that the chromosome had been around for maybe 200, 300 million years, this was a fairly short order, short period of time being projected. And that model was based on the idea, that projection was based on the idea that the Y chromosome had been losing genes at a steady drip over those 200, 300 million years.
But it sounds like it was an assumption.
It was an assumption. And what we've found in recent years, by comparing the Y chromosome of the human with the Y chromosome, especially of the chimpanzee and of the rhesus monkey. So we're separated from chimps by 7 million years, from the rhesus monkey by about 25 million years. By studying those, and more recently some additional mammals, we've been able to reconstruct the trajectory. That's how you did that. The trajectory of gene loss on the Y chromosome. And what we found is that most of the gene loss on the Y chromosome occurred very early.
Okay.
So it was a
Well,
calamitous.
That was a big find.
Yeah, that was exciting. It was a calamitous, precipitous loss of genes, and then the Y chromosome sort of leveled out and has been sailing along pretty happily with a small but stable set of genes for actually on the order of about 100 million years.
But it sounds as though because there was so much on the X that it didn't need, so that it was a very healthy, shall we say, strategy on the part of the Y. I mean, it was a very good adaptation kind of a thing. So it wasn't anything...
It evidently didn't cause that much trouble.
Right.
But actually, we're very intrigued now, and maybe we can turn to it. Very intrigued by the set of genes that the Y chromosome actually hung on to.
Yes.
And for a good while, the other piece that fed into this understanding, this sort of prediction of the Y's imminent demise, was that the loss of genes from the Y chromosome had been sort of a willy-nilly, random exercise. But what we've come to understand very recently is that the genes that the Y chromosome hung on to, it hung on to very purposefully, in a sense. And we think that there are a small number of genes that persist today on the human Y chromosome, shared with the X chromosome. We think there's a small set of genes for which it's quite important to the individual to have 2 copies of these genes.
Oh, how interesting.
Okay? So that women with 2 X chromosomes have 2 copies of these genes, and men with 1 X and 1 Y also have 2 copies of these genes. We think there's a very small set, maybe as few as a dozen genes, that are shared between the X and the Y, where having 2 copies is critically important.
That is amazing. It seems really that those were smart moves on the part. But the other thing there is that could you even have figured this out like even 20 years ago? Or maybe, I mean, was modern sequencing, did that give you the ability to see this?
Well, so great question. So the only reason we've come to understand the Y chromosome at all is through the era of genomics and the era of gene and genome sequencing. So what's now clear is that almost everything we thought about the Y chromosome before the era of genome sequencing was wrong.
Yeah, I see.
We can and so because the Y chromosome, and part of the explanation for that is because the Y chromosome is present, is passed from father to son to son to son to son, all the tools of Mendelian genetics, all those things that Mendel taught us, and that anybody who ever takes a genetics course learns, those things apply to genes and chromosomes that come in pairs, one from mom, one from dad. The Y chromosome doesn't follow those rules, so it could never be understood through the standard tools and ways of thinking of genetics. So it absolutely had to wait until DNA sequencing
really
came along. And even as the era of genome sequencing opened up, it was about 12 years ago, we started to get a handle on the sequence of the human Y chromosome. We thought when we had the sequence of the human Y chromosome, we were so excited. This is telling us so much. But then it's more recently when we've been able to add the sequence of the Y chromosome of other primates and in the very recent past to begin to see the Y chromosomes of other mammals. We get so much richer and deeper a picture. We can get a look back into the past of the Y chromosome. And so all these notions about the Y losing genes rapidly and then stabilizing, it comes not just from a study of the human Y chromosome, but from the ability to compare our Y and X chromosomes with the Y and X chromosomes of our sister-brother species.
Right.
Yeah.
Let me get this clear then. So what you've seen, and now you can sort of verify it, is that this loss occurred at least across mammals very early. So it's the same thing. That Y did the same thing. It didn't matter so much the species. It mattered that it was the Y, and they made that.
Well, it's a great point. So let's understand that actually the X and the Y, our X and Y chromosomes, had their beginnings before placental mammals even arose, or as they were arising.
Right.
So what I mean is, our X and Y chromosomes are found in altered form in essentially all mammals.
Okay.
And so what I'm saying is the evolution of the X and the Y from an ordinary pair of chromosomes was actually a feature of the whole, what would be called the radiation of placental mammals.
Right. That just
so the beautiful thing is that if you're seeking to understand the whole, all of mammalian radiation in its great glory, and you want to understand the sex chromosomes, these are completely intertwined subjects.
So you expect to see this then as you investigate more and more mammal species.
So we're now studying in great detail. Actually, we wish we could do more, but we're focused on 8 mammals. So we're studying the human, the chimp, the rhesus monkey, the marmoset, the rat, the mouse, the bull, and for good measure, one marsupial, a South American opossum.
Okay.
And then we actually add the chicken as a bird, as an outgroup to mammals.
Yeah.
So it turns out that, again, all of these mammals and many, many more share, to a first approximation, they share our X and Y
That's
chromosome.
amazing, yeah.
But in some cases, we've been split from some of these other mammals for 80, 90, 100 million years.
Yeah.
So their X and Y chromosomes have specialized away from ours a bit. But it's all one grand experiment of nature.
Yeah, that's amazing. Let me ask you, now when you're doing this sequencing it sounds from a lay point of view that, hey, this is a piece of cake. Now, just put it in this machine and get a reading.
Sure.
But when you started this, and you were right there at the beginning, I think, it was evidently very difficult to do. What has changed? Why was it difficult, and what's changed?
It actually remains difficult.
Oh, I see.
To
Just
sequence
the Y chromosome.
So it turns out that yes, today the sequencing technology has evolved, has improved to such an extent that we'll now talk about, sequencing someone's genome for medical purposes. Not quite at the drop of a hat, but we're getting there.
It's getting very close fast.
But there's something, this is a somewhat different thing. Here we're talking about generating the sequence of the Y chromosome of the human or of another species for the first time.
Oh, I see.
So actually, what we've become really good at in medical genetics is resequencing the human genome.
I see.
But it turns out that establishing the reference sequence of a Y chromosome is particularly difficult because, as we found, we didn't know this before we got deeply into it. It turns out that Y chromosomes carry massive mirror images of their own sequence. So, for instance, the human Y chromosome carries 8 very large palindromes.
Oh, for goodness sake.
So, a palindrome is a sequence that reads the same. Mirror. So the palindromes on the human Y chromosome can be incredibly large. The largest one is 3 million letters in length. 1.5 million letters.
Yeah.
And then another copy flipped around in the opposite direction. And the standard methods of sequencing, which involve taking our DNA and chopping it up into little bits, sequencing all these little bits, and then asking a computer program to put it all together.
Yeah.
Things like these palindromes completely befuddle the standard methods of assembling a genome sequence.
I see.
And what this has meant is that it has required a much more painstaking approach and a much more sort of deliberate and maybe compulsive, I should say, approach to actually accurately assemble the Y chromosome.
Yeah.
But of the human and actually of the other species.
Yeah,
So it's required
It's just
an
this.
extra level of investment, an extra level of work. And this helps explain why, actually, by today, we have a rough cut sequence of the genomes of many dozens of mammals. But we today have published the Y chromosome sequence only of 3 primates: human, chimp, and
because
rhesus.
it's
We're
still
way
so
behind.
difficult. It's
we're
so
still
difficult.
behind. Yeah.
Right.
We're working to pick up the pace now.
Well, at least now we understand why this has been such a challenge. And I was thinking that it was because initially the whole mess was a challenge, but then there was some extra difficulty around the why. But there is.
Well, there were a couple of things going on there. So one was, the whole. At the outset, sequencing genomes was a very hard thing.
Right.
Then, as I mentioned, in the case of the Y chromosome, it is inherently a more difficult target to sequence than the other chromosomes. But then there was another, for a variety of reasons, when the genomes of new organisms were being sequenced, the decision was taken to sequence the female genome. Actually,
because it was easier?
Well, because there were a number of reasons.
It was prettier.
First, it was assumed, it was actually understood, the Y had been under such sort of negative publicity for long, it was actually assumed that it wasn't worthwhile sequencing the Y chromosome. I'm not making this up. And so
science is so embarrassing.
So part of the rationale was, well, let's just do the female because we don't really want the Y. I mean, it won't really add that much anyway, will it? And I'm not making this up. But there was another reason, which was that the methods for sequencing the human genome were designed to, with the realization that for most of the chromosomes in our genomes, there would be 2 copies in the mix.
Yeah.
And so there was a statistical sampling method that was put in place that said, I'm going to ask that you have 2 copies of the chromosome. I will fold them in as if they were together, as if they were one, and I will call that the reference sequence.
I see.
And so the decision was taken to sequence female genomes because then you'd have 2 copies of the X chromosome.
Right.
And if you'd sequenced a male genome, you would underrepresent the X and the Y. You wouldn't get a decent coverage of either.
Right, so that was the excuse.
So there were a whole set of reasons.
Yes.
So in other words, it's taken a dedicated add-on effort to capture the Y chromosome.
Right. I see that now, but I'm amazed that it took a long time to get past some of these obstacles and also the thinking.
Well, and I would say these mental obstacles are actually still there, and it's why I say I've had so much fun sort of traveling with the Y chromosome in the face of all of these thoughts that it actually wasn't worth examining.
Right. Well, you were sort of out there alone too, weren't
Pretty
you,
much
for a long time? But now they're more respectful, right? It's all in vogue, but it just changed very quickly. But it's a wonderful story of science, of a correction mechanism that people go along for a number of reasons with certain assumptions, and whammy, you're completely wrong. And you have very prestigious journals, telling you what's right one day and then it's wrong.
Absolutely.
So you have had a good time with it then.
Well, it's been, I've felt really privileged to travel with the Y, and then.
But you took a chance.
Yeah. I mean, there were, I've been traveling with the Y chromosome for, 30, 35 years now, and it's a lot of fun to look back at what did people say about the Y chromosome
Yes.
30 years ago,
Yeah.
right? And it wasn't very favorable.
Right. And even recently,
it was thought. So it's actually very interesting. If we take the clock back 2 or 3 decades,
right.
it was generally assumed then that there was one function of the Y chromosome, maybe one gene, that was involved in directing the fetus to develop as an anatomic male.
And that's
not as an
all.
anatomic female.
Right.
That was widely understood to be the only function of the Y chromosome. And so what has come to light in recent decades is this incredible specialization of the Y chromosome. Well, I should say, first of all, that original surmise is correct. There is, in fact, a gene on the Y chromosome that plays a pivotal role
but it's not
in
the
sex
whole
determination. But in addition, there are many genes on the Y chromosome that are involved in sperm production.
Yeah.
But then what has, I'd say, come to light most recently is a set of a dozen genes that are shared with the X chromosome, that are actually expressed, that appear to be active in all the tissues and cell types of the body, and that have no obvious connection to sex differentiation as classically taught in medical schools.
Right.
And
still in medical schools.
Yeah. So...
and that is really new. I hope you don't have the big battle there, but I believe that has been the source of your interest in these male-female biased diseases.
Absolutely.
Okay.
Absolutely.
We have some other things to get into.
Sure.
But let's talk about that, because that's a new wrinkle, I believe.
Yeah.
I don't know. How long has that been out there? I mean, we all know there are disproportionate...
So there are many diseases. You know, it's actually almost the rule, not the exception, that even for diseases outside the reproductive tract. So let me back into this and say, you know, obviously things like prostate cancer, that affects men.
Right.
Breast cancer primarily affects women. Uterine cancer affects women. So we could go down the reproductive tract and think about disorders that affect one sex or the other for obvious anatomic reasons. But then there are many diseases outside the reproductive tract that affect tissues and organs that are common to men and women, many diseases that occur much more frequently or more severely in males than in females, or in females than in males. And medicine has known about this forever, but it isn't it hasn't been a closely examined question. And actually what I find most interesting is that there's been very little thought given across the sweep of human disease about this question in general. In other words, so let's take I could mention the autoimmune diseases. Rheumatoid arthritis, much more common in women than in men. Lupus, the same thing. Or I could flip around and say autism or autism spectrum disorders, much more common in boys than in girls. So we're just now beginning. And when I say we, I mean the whole biomedical enterprise. We're just beginning to actually name this question and to approach it a little more systematically. So I believe that each of these diseases that showed a sex or gender bias was seen in isolation.
Very.
And you see what I'm saying is, well, maybe you'd say, let's take the autoimmune disorders and think about them as a group. But what I'm suggesting is that we now need to take this whole sweep of disorders and diseases that show strong sex or gender biases in incidence or severity, and we need to begin to disentangle the underlying causes of these biases. And so I think there are many We don't have any. We have very few, if any, answers today.
Right, but you're asking the question. It sounds like that's the important thing, because it's for the first time that that you're out there saying why.
Yeah. So I would say the prevailing view, when I talk to my physician colleagues, and I've got lots of them in different areas of medicine, when I ask them, what causes these sex or gender biases in the diseases that you study or that you care for? The answer I almost always get is, I don't have a clue.
Right.
We
got that far. They don't know a clue.
If I press harder, the answer is usually, it could be the sex hormones.
Yes, so it has to be the hormones.
Well,
but you went further than that.
But what we begin to wonder is, yes, I have no doubt that the sex hormones, estrogens and androgens, play important roles in these sex and gender biases in disease incidence and severity. But we begin to wonder whether some of these genes that are shared between the X and the Y chromosomes, whose significance was not appreciated until very recently, and I mean this year.
Oh, this year, wow.
And when we recognize that these XY gene pairs are actually expressed throughout the body, so we begin to say, oh my gosh, it could be that even the cells of our skin,
your toenails,
the cells of our gut, at some level, they may know whether they're XY or XX. And perhaps that contributes to these very different predispositions on the part of males and females to a wide variety of diseases outside the reproductive tract.
Well, let me ask, maybe it's still very new, but this is another thing. Do you see this in other species as well, even in our ape relatives? Would you see this difference between male and female diseases, if such a thing exists, or is it just us?
I think it may well exist. I would be surprised if it doesn't. It certainly exists in mice, in laboratory mice, where you study various disease models are created and studied in mice. There can be enormous sex biases there as well. So I think it would be very surprising if it were not the case across the entirety of the animal kingdom.
Right.
It's just that we, of course, are most concerned about our own health.
Well, and those are the things when we talk about diseases, we mean us too. Yeah. But the other thing is, it seems like that really buttresses the argument that we need to be looking at this genetic thing, at these chromosomes that are playing a very large role, because from the public view, we hear hormones all the time, and that's one of the questions I was going to ask you, is that we have these endocrine disruptors that are ubiquitous now, and we're seeing sex ambiguity and stuff in, say, reptiles and amphibians and so on. But if we pursue this, that's hormonal. How does that affect what you're talking about, the disease, sort of like that? Exactly. If you had from the hormonal argument, you'd say, well, look for autism in reptiles or something.
Well, so we have this explosion of information and interest in the question of environmental endocrine disruptors and so on. What I would say is that we need so much more basic, fundamental research done in that space that I can't. So it's almost, I think actually our public storytelling in that space is actually, it has foundation in the scientific literature. But the scientific literature in that space is so meager. We need so much more research in that arena. And the whole question of the endocrine disruptors and such is it presents questions of bewildering scientific complexity because of the number of chemicals that we as a species have introduced into our environment.
Right.
And probing how each of those many, many chemicals might disrupt our systems is far beyond the capacity of the very limited corner of the scientific enterprise that's devoted to those questions at present. What is striking, though, is how fragile our capacity and the capacity of our species' capacity and of other species' capacity for sexual reproduction is to environmental disruption.
Yeah.
What I'm saying is that there's something about the mechanisms of sexual reproduction that make them very vulnerable to chemical perturbation.
Right.
That's a commonality that is emerging. But why that is and how it plays out, we really...
Well, I understand.
I don't want to pretend that we know more than we do.
Right.
I think we're just, again, beginning to scratch the surface of those questions and how this all works. And, of course, with an eye towards, well, what can we do about this?
Well, that's true, but also then the question going back to these male or female prevalent diseases, what will that mean if you have this additional factor which is so pervasive right now? So we're not necessarily seeing that, I guess, in humans so much. But
I would say that these two, right now I would say that these two sort of storm systems have not yet collided intellectually.
Okay.
So what I'm saying is one storm system is why so many diseases outside the reproductive tract with a strong male or female bias.
Right, and that's where you're at. Right.
So that's one whole set of questions. Another set of questions is what is happening in our environment that is disrupting the sexual reproduction of so many species.
Right.
And in many cases, we actually look to other species, actually even including alligators, with temperature-dependent sex. The disruption of sexual reproduction in these many species is sort of serving as the canary in the environment to raise our own concerns. But what I'm saying is these two storm systems, sex bias in disease and environmental disruption of sexual reproduction, these have not yet, to my knowledge, have not yet intellectually collided. In the way that you're suggesting.
Or converge.
Maybe they will. So we'll have to see in the future what that looks like.
So, but that's not a part of that. Your concern immediately is not that you are taking the classic male-female.
I'm very concerned about both of these. And I'm very interested in both of these developments.
Right.
But I, for one, don't have a way of thinking about them together.
Yes, I understand. But in terms of where are you now with looking at the male-female, what shall we say, biased diseases or conditions? Because they're syndromes, they're conditions, they're diseases.
So we're barely out of the starting blocks.
Okay.
So, but we're trying to figure out, actually, how to approach these questions. So we need new tools, we need new ways of thinking, and what I'll say, one of the ways we're beginning is to think about simply cataloging the molecular differences between males and females throughout our bodies.
Yes.
So I like to make the analogy back to the time several centuries ago when the early anatomists began to dissect human cadavers.
And they were surprised.
And discovered that we actually had a circulatory system. We had a heart and there were all these pipes coming out of it and things like that. And said, this is how it works.
Right.
Well, guess what? Almost all of those early dissections were done on male cadavers.
How about
then
that?
Somebody dissected a female body,
and
they were shocked, saying, "Oh, something is wrong." And so I would say that where we are today with respect to our molecular inventory is where the anatomists were before they'd actually dissected a female body.
Well, let me ask this then, is that when you were working on the why, you really, I think, were a pioneer out there sort of on your own. You didn't have an army of people saying, yes, this is right. In fact, there were detractors. But now with this, and something that's been just ignored, really, I guess, in terms, is it as difficult to convince people that we need to look at this chromosomal factor in terms of these diseases?
So I've really only been talking with colleagues in medicine and in science about this within the last year or 18 months.
So it really is new then.
It's very new, but I would say the reception has been exhilarating.
Oh, wonderful.
I mean, people are excited about this idea, and and I think actually the time is ripe to get serious about trying to understand and actually trying to understand these sex biases. And it's worth recognizing it's against a backdrop of now, in this era of genomics, there is much talk of personalized medicine.
Yes.
Okay. Well, it turns out, so in thinking about personalized medicine, we're thinking about how can we learn from the sequence of someone's genome, together with other information about diseases at which they might be at particular risk, and about which we might do something in a preventative sense, for instance. But I would argue that actually the way we've had a unisex model of personalized medicine.
And that will now change.
And I think that absolutely has to change. When, in fact, the single greatest identifiable risk factor for many diseases is, are you a male or are you a female? We need to actually acknowledge that and take that into our thinking and see what we can make of it. So there may actually, for some of these diseases, the disease in males and the disease in females may, in some sense, be different disorders.
Oh, that is interesting.
Even if we give them the same name.
Exactly. How interesting, which you would uncover as a result of this, because it just sounds like a whole new direction. It does come at an opportune time when there is this sense of we need to personalize medicine much more, and it's within our capacity, certainly in the future. But what an exciting prospect this time. I'm glad we got to meet you before you get the Nobel.
That's very kind.
We have to stop there, and I would know people would like to ask you a few questions.
Sure.
So I thank you very much for joining us tonight.
Thank you very much. It was wonderful.
Yes.
Lots of fun.
Okay. And I will let people just go ahead and talk with you.
I have a question.
Yes.
My name's Regina, and I see what Whitehead a lot.
Yes. Hi, Regina. All right.
Does the sequencing provide any light on some of these 12-ish genes that shares with the X? Any general categories, for example, kinases or something? Any idea
what these genes are?
Might be?
Well, so it's very, very exciting, Regina, because it turns out these, if there is a broad theme among these 12 genes, they look to be global regulators of gene activity. Okay, so they include chromatin modifiers, transcription factors, translation factors. So these are what it really blew our minds when we began to see this sort of commonality among them. So it looks like they, we describe them as global regulators of gene activity.
That makes sense.
Okay, and it turns out that actually the X member of many of these XY pairs, in some cases the X side of the pair has already been implicated in some diseases. So it's the Y side hasn't been looked at so much, but what I'm saying is these 12 XY gene pairs are very, very, from a modern biologist's point of view, these are very sexy pairs. They're really, they have a lot of name recognition already. The X genes already have name recognition, already have developing literatures. And so it's very exciting for us because in some cases, some of these XY gene pairs we had discovered as long ago as 17 years ago. But in the ensuing 17 years, the cell biologists and the biochemists have been hard at work telling us what these gene pairs do, what the products of these gene pairs do. And so it's when we come back and put it all together and look at it now, these are a very exciting pair of genes.
Not surprising, actually, but exciting.
And then I think this actually contributes to the enthusiasm. It's actually so surprising. I finally have something which meets with an almost immediately enthusiastic response. And people, I think, and it's actually very gratifying because for so many years, the view was that this research into the Y chromosome, it has, it's of some, it's sort of curious. But it's a rather arcane subject, right? A very limited significance. But once you start saying, oh, how about lupus? How about autism? And you start saying, oh, you mean all of medicine might be the playground now for thinking about this? It completely changes the way people think about it and receive the information. Yeah. Sorry.
12 shared genes, are they published somewhere that I can look at?
Well, yes. We actually just published, and I should have sent you the preprint. That's right.
3 papers in Nature or something?
Well, no, we just published an article in Nature earlier this year. And yeah, I think it was in April. Late April, yeah.
My question is, there's something some more for you to do. Have you looked at the regulatory regions of these 12 genes, and are they the same in the X and in the Y?
Fantastic question, right. So that is, in fact, one of the graduate students in my lab is now looking at precisely that question. So the interesting thing is now, if we think about these 12 XY gene pairs, the question becomes, are the products of these genes identical in function? Do they function equivalently? But maybe even if they function equivalently, maybe they're not expressed in quite the same way. And so we're very interested in the possibility that evolution has resulted in slightly different regulations of the X and the Y flavors of these genes. Really great question. Yeah, that's right at the cutting edge. Yeah.
I have a question. If some disorder in the sequence, they will cause diseases. If
Yes.
you take the medicine, it will put the sequence in the right order or kill some virus or something.
Let me make sure I understand. So you're saying if, in some cases, an alteration in the sequence of these genes causes a disease,
Yes.
then were you asking how should we correct Well, okay. So, I think there is a lot of excitement in just the last year or two around new genome editing methods.
Protein engineer or molecular engineer?
No, actually, DNA. So some methods for actually changing the spelling or maybe correcting the spelling. Now, these methods have in the last 2 or 3 years, actually in the last year or two, become all the rage in modern biology. They've been incorporated into many experiments. People are just beginning to think about and just beginning to apply these to the possibility of correcting defects responsible for human disease. But I think we're going to be hearing a lot more in the near future about actually correcting spelling errors. Yeah.